we've finished with the opisthokonts. We are then going to move on to the rhizarians. So Rhizaria is this relatively diverse group of protists, and what we're going to see is that the DNA, the genetic evidence, is why these are all put together in the same group, even though they have quite a bit of diversity in the overall body morphology. So this is one of those groups that maybe we think is a little questionable, but genetically they seem to be related to each other. Many of these fall under the group of being kind of amoebas. We already saw that amoebozoans had a lot of amoebas. These amoebas are a little different because they have pseudopodia that are long and thin. So in the picture here, these are the pseudopodia sticking out here. These really long, thin, thread-like pseudopodia. So remember the amoebozoan pseudopodia were kind of wide and were shaped, more like a tube or a lobe. These are more like threads or like hair-like pseudopodia, real thin. There are three different groups of rhizarians. We're going to start out with the foraminiferans. Now, because foraminifera is such a long word, we often will reduce this to just the forams. The name actually does mean something, though. If you've taken BIO203, you've probably learned about foramen and foramina. It basically just means whole. Fer, F-E-R, is a root word that means bearing. So foraminifera just means hole bearing. They got holes in them. All right, the foraminiferans have these holes in their shells, or sometimes we call shells a test. So a test with CaCO3. CaCO3, that's calcium carbonate. Same stuff as limestone or chalk. So foraminiferans are made of chalk, basically. The foraminiferans are all single-celled, although there are some pretty big ones. There's some that might be a few centimeters, like an inch in diameter, which is pretty large for a single-celled organism, very large for a single-celled organism. The foraminiferans are known for some things, one of which is that they have a mutualistic relationship with an alga that lives inside. So the alga is able to do photosynthesis, provide, sugar for the host, and then the host gets that sugar and provides a place to live for the foram. Foraminiferans are known for a couple things, one of which is they are really well represented as fossils. And that representation as fossils all over the place, all around the world, in lots and lots of different sediments, it's really useful because it can help us to age things. So, for example, let's imagine that you had an organism, a fossil of an organism from, let's say, South America. And then you had another fossil organism from Africa. And let's imagine that they were, I don't know, some sort of mammal type of thing. And they seem pretty similar to each other. And you were trying to decide whether they were actually related or whether those similarities were due to convergent evolution. That maybe they weren't closely related at all, but they just looked similar. Well, one of the ways you could help to decide that is to figure out whether they had been around during the same time. Were they living contemporaneously, that is to say at the same time, or are they from completely different time periods? And one of the ways you might be able to figure that out is by seeing what sort of foraminiferan fossils there were in the same strata. That if they were living at the same time, you should find the same foraminiferans. But if they are different foraminiferans, then maybe those were living at different times. And so that would help to make a decision about whether those are closely related to each other, because you could help to figure out, at least relatively, what the ages were. Foraminiferans are also used as indicator species. So indicator meaning they can tell us something about conditions. So different species live in different conditions, right? Warmer water versus cooler water, for example. And so by knowing which foraminiferans were found in an area, when you're looking at those fossils, you can have some idea what that climate was like, what those conditions were like. Okay, our second type of rhizarian then are the radiolarians. Look how cool these things are. So the picture on the right is actual radiolarians. The picture on the left is actual radiolarians. On the right is a painting of radiolarians. When microscopes got to be high enough quality that we could see these things well, people were really impressed by them. And there were actually paintings that were done that people would hang in their parlor as discussion pieces because they were such spectacular and incredible organisms. The reason they look so cool is that radiolarians have shells made out of silica. So basically they were glass. And you can see they're just amazing, intricate details and really, really spectacular looking. Radiolarians have those long, thin pseudopodia, and those pseudopodia are reinforced by microtubules. Now, if you've taken BIO201, hopefully you remember microtubules. Microtubules are one of those cytoskeletal elements. They're basically little tubes. And so they are these long, thin, you could think of them as like, the cardboard tubes out of wrapping paper, but very tiny, that reinforce those long, thin pseudopodia. And then also in those pseudopodia, there's a layer of cytoplasm. Because that cytoplasm is always moving, that means any food particles that get caught by those long, thin pseudopodia, those food particles get basically washed back down into the main part of the cell where they can be consumed. All right, so foraminiferae, radiolarians, haha. And then my favorite group of rhizarians, the cercozoans. Now, if we take a look at the term cercozoa, I like to put my own kind of interpretation on it. Zoa is a suffix that means animal or animal-like. Cerco probably means something. I like to think of it as circus. So, these I like to think of as the circus animals. And you'll see why. It's because they're so weird. Before we talk about the weird ones, let me point out most cercozoans are heterotrophs. All right, so most of them are capturing food, but the real weird ones we're going to talk about are not heterotrophs. But most of them are. Don't forget that. However, one that's not is a species in the genus Pollinella. Now, this is a picture of Pollinella, and probably you'll notice pretty quickly it has that green. Pollinella is able to do photosynthesis. And it's able to do photosynthesis because of a completely different primary endosymbiosis than all the rest of the eukaryotes. All right, so here's what I mean. Here's our phylogeny, right, of all the eukaryotes. The ability to do photosynthesis amongst eukaryotes can almost exclusively be traced back to a primary endosymbiosis that occurred in the Archaeplastida, right? What's archae mean? Like archaeologist? Archae means old. So Archaeplastida, old plastid. So amongst the Archaeplastida, we had this primary endosymbiosis. And then, through secondary endosymbioses and tertiary endosymbioses and quaternary endosymbioses, that ability to do photosynthesis was distributed throughout quite a few of the eukaryotic supergroups. That photosynthetic ability can be traced back to this old plastid, if you will, in the Archaeplastida, which has then been distributed through later secondary, tertiary, and quaternary endosymbioses to the rest of these supergroups. Except right here in the cercozoans is a different primary endosymbiosis. That Pollinella gained its photosynthetic ability from a different primary endosymbiosis than gave rise to the photosynthetic ability for all of the rest of the eukaryotes. That there were only two primary endosymbioses of photosynthetic organelles. One occurring in Archaeplastida and giving rise to photosynthetic abilities all over the place. The other, a more recent primary endosymbiosis occurring in the genus Pollinella. And not all species of Pollinella can even do this. It's just one species of Pollinella that has this different primary endosymbiosis. It was so recent, in fact, that the cyanobacterium that's living in it as an endosymbiont inside of Pollinella still has some of its peptidoglycan. completely different endosymbiosis. Primary endosymbiosis, right? It's not unusual to have other endosymbioses that occur. We see lots of them across the eukaryotes, but this is a different primary endosymbiosis. All right, let's look at some more circus animals. Another one is this group known as the chlorarachniophytes. We've already seen this root word chlor. We said it meant green. We've seen phyte, means plant. Hopefully you remember that. That's a good one. Plant. Arachnio, spider. Green spider plants. The chlorarachniophytes we could refer to as the green spider plants. Now, I'm not sure if that's actually accurate, but that's what I like to think of them. And you can see that that green coloration comes from the fact that they are able to do photosynthesis, and it's a secondary endosymbiosis. A secondary symbiosis specifically of a green alga was what was engulfed. So, what I'm saying here is, within the cercozoans, we have the ability to do photosynthesis in a couple different groups, at least. One of them, a primary endosymbiosis, right, of a prokaryote, of a cyanobacterium, and then the other, a secondary endosymbiosis of a green alga. That green alga, of course, who got its photosynthetic ability from a primary endosymbiosis that happened towards the beginning of the Archaeoplastica. The green spider plants are mixotrophs, so in addition to being able to do photosynthesis, they can also consume food particles. Probably that's why they have those long, thin pseudopodia all connected together there in what looks kind of like a spider web, is capturing food particles. And then if they can't catch anything, they can also do photosynthesis because they're green. Really cool group of organisms. All right, so that only leaves two supergroups left. We're going to move on to the chromalveolata. Now, we're going to have a story here that sounds similar to some other things we've heard. Amongst the chromalveolata, there's genetic evidence that suggests that the chromalveolates all belong in this same supergroup, right? That the genetic evidence shows that these are related to each other. We think that this supergroup chromalveolata was first started by a secondary endosymbiosis, specifically a secondary endosymbiosis of a red alga. However, this supergroup is controversial, and here's why. We think there was a secondary endosymbiosis of a red alga, but some of the members of the chromalveolata don't have plastids. All right, well now it's possible for organisms to lose plastids over time. If they live someplace where there's no sunlight, for example, a photosynthetic organelle is not helpful, and so maybe over time some individuals lose it, and then because there's this evolutionary advantage to not have to expend energy on it, they get selected for. And so it's possible for plastids to be lost. We've seen it happen in other lineages. But not only are there no plastids, there's not even any leftover plastid genes anywhere in some of these. And so we have this kind of difficult situation where we think that there's a supergroup that is derived from a secondary endosymbiosis. But in some groups, we find no evidence of that secondary endosymbiosis. No photosynthetic organelle, not even any leftover DNA from that potential historical photosynthetic organelle. So we're going to consider these two as belonging to the same supergroup here, but we're just going to know that, who knows, maybe we're missing some stuff. Maybe these aren't actually related to each other, but genetically it looks like they are. All right, so let's focus in, though, within the supergroup chromalveolata. We're going to start with this subgroup of the alveolata, the alveolates. The alveolates, we are pretty sure, are each other's closest relatives. It seems like that these three groups are most closely related to each other. And this group actually has a name that we can trace back to a phenotypic characteristic. So the alveolates are named that because they have alveoli. What are alveoli, you might ask? Well, alveoli are these little membrane-bound sacs. You might have heard the same term is used in lung tissue. Again, little membrane-bound sacs where gas exchange occurs. Well, here we have these little membrane-bound sacs that are around the outer plasma membrane of the cell. All right, so here's the membrane. We've zoomed in on a cell here. So this is one cell. Here is a flagellum coming off of this one. And then we have these little alveoli, these little membrane-bound sacs that are going around the outside of this cell. So this is the outside of the cell, this is the inside of the cell. Here are these little membrane-bound sacs. The alveolates all have them. It gives us a useful characteristic to name them after. What's it used for? What's the benefit of little membrane-bound sacs? Well, it could be used for, I don't know, storage maybe, or protection. We don't know. Good question. If you find out, let me know. We don't know what they're used for, but we do know that all the alveolates have them, and it provides us a way to differentiate them from other protists. All right. We're going to start amongst the alveolates talking about the dinoflagellates. The dinoflagellates get their name, again, from a phenotypic characteristic. I always like to ask, and unfortunately, since we're not actually in the classroom, you don't get to answer me. Was anybody really into dinosaurs as a kid and actually knows what the word dinosaur means? If you do, dinosaur means terrible lizard. So are these the terrible flagellates? They're not. This actually comes from a different dino root, and this one means spinning. So these are the spinning flagellates. They might spin because they have two different flagella that are in grooves on the cell. And so if you watch them swim through, they spin as they swim. All right, the spinning flagellates. What are they known for? Well, they're primarily known for the fact that they lead to this thing that is known as a red tide. You might have heard of this if you've been in coastal areas. We call this a bloom. A bloom just means a whole bunch of them are there at once. So lots and lots and lots and lots and lots of dinoflagellates results in this algal bloom. We call it red tide when it's dinoflagellates. It's not really the best name for it. One, because it's not always red. You can see some of that's orange and it can be yellow. It doesn't really have anything to do with the tide. But red tide is what we call it. And red tide is really kind of dangerous because these dinoflagellates have toxins in them. And so, especially if, I don't know, let's say that you're eating some shellfish. It would be very shellfish of you. You're eating some shellfish from an area where there's a red tide. Shellfish tend to be filter feeders that filter a lot of things like toxins out of the water. And there ends up a concentration of toxins in those shellfish. And then, if you eat shellfish, you end up with even a higher concentration of toxins in you. This is an important concept in biology known as biomagnification. The idea that things that are in the food web become higher and higher and higher in concentration. Because, right, you eat multiple shellfish, and those multiple shellfish ate many dinoflagellates. And so you end up with lots and lots and lots of that toxin concentrated in your tissue. And that's bad for you and could kill you. So you have to be real careful with that. It can also kill fish in the water. These red tides are pretty serious. Sometimes if you go to the beach to go swimming, there'll be a sign that says no swimming, red tide. I'll point out that the red tide comes from pigments called carotenoids. What do you think those pigments are used for? That's right. Photosynthesis. Photosynthesis. So, dinoflagellates, pretty cool, can be dangerous, but look how pretty that is. Our next group of alveolates are the apicomplexans. Now, apicomplexans, their name does mean something. Apa comes from an apex is just like an end. In this case, that end has a complex for burrowing into things. So, if we take a look at this picture, we can see some apicomplexans that have burrowed into things. Specifically, members of the genus Plasmodium cause malaria. So our picture here is primarily showing a bunch of red blood cells. But inside some of those red blood cells are some Plasmodium. And Plasmodium are the causative organisms of malaria. They've got into those red blood cells by burrowing their way in with that complex at their apex. Now, I'll mention that this is a good strategy, right? Because if they didn't burrow their way in, well, the immune system might get rid of them pretty quickly. But once they get safely inside of those cells, the immune system doesn't even see them. Let's take a little closer look at what a Plasmodium would look like. So here's that complex at the apex. It's going to burrow its way inside of the cell. And here, don't be scared, this is the life cycle of Plasmodium that causes malaria. So I want you to look at it for a second. And I want to ask a question. Does this Plasmodium have sexual reproduction? If you said yes, you're right. How do we know? Well, if you look down here in the bottom right-hand corner, we have meiosis. And then, over here, we have fertilization. All right, so let's look and see what actually happens in this life cycle. So, we'll start at number 1. A mosquito bites you. And in so doing, you get some Plasmodium from the saliva of that mosquito. Once inside your body, the first place it goes is to the liver. Liver cells get infected. The Plasmodium starts to multiply inside the liver, and then it leaves the liver cell and goes into the blood. Once it gets into the blood, this is when it's going to burrow its way inside a red blood cell. Now, it can do this for a long time. There can be lots and lots of Plasmodium inside the red blood cell. And then, at some point, this bad thing happens. The red blood cell bursts open because of all the Plasmodium inside of it. When we have a cell burst open, it's called lysis. So the cell lyses, breaks open, and releases all those little Plasmodium from inside of it. This is what causes the symptoms of malaria. Red blood cells exploding, it turns out, is bad for you. Once released, the Plasmodium can go through meiosis, produce gametes. Those gametes can be picked up by another mosquito that would bite you. Once inside the mosquito, those two gametes come together, fertilization takes place. Fertilization results in a zygote. A zygote we're just going to think of as a fertilized egg. That zygote goes through mitosis, ends up in the saliva of the mosquito, and we start the process over again. It's this disease-causing part, when the red blood cells explode, we don't necessarily understand exactly why they choose what times to explode they do. Different types of malaria might happen at different times, but it's pretty detrimental, as you could probably guess, having your red blood cells exploding. Because what do red blood cells do? Carry oxygen, and oxygen is pretty important. All right, let's move on then to our last type of alveolates. These are the ciliates. Paramecium would be a good example of a ciliate. We see that they get the name ciliate because they are completely covered in cilia, lots and lots and lots and lots of cilia. On the picture on the right, the cilia just kind of look like a fuzziness around the edge. There's so many of them. Ciliates have this really weird life cycle. And you can see, the first thing one of the things you might notice as you look at the diagram here, is that ciliates have this situation where they have two different types of nuclei inside the same cell. Macronuclei is big nuclei, and micronuclei is smaller nuclei. Well, why do they need to have two different types of nuclei? What are they used for? Well, let's take a look at the life cycle here. Remember when I said that some protists don't have a sort of a normal life cycle? This is a good example of that. All right, during normal everyday cell function, a macronucleus is directing things. At some point, the cell says to itself, I would like to go through sexual reproduction. Let me find another cell, another paramecium, that would also like to go through sexual reproduction. These two paramecia then form this connection between them, this little corridor. Each paramecium at this point has one macronucleus and one micronucleus. Now, after this connection, known as a cytoplasmic bridge, is formed between the two paramecium, then the micronucleus in each cell is going to go through meiosis. So that must mean that the micronuclei were originally diploid. Because they're undergoing meiosis, they must be diploid. The resulting nuclei that are formed then are haploid. There are 4 of them, but 3 of them disappear. They disintegrate. So we're back to having a macronucleus and a micronucleus, but now the micronucleus in each cell is haploid instead of diploid like when we started. At this point, each micronucleus goes through mitosis, so we end up with 2 micronuclei in each cell. And then, here's the sexual part, the 2 paramecium trade 1 macronucleus. So, we now have, each paramecium has a macronucleus and 2 micronuclei. The 2 micronuclei, one originally from the cell, one from the other cell, now fuse together, that is to say, fertilization, forming a diploid micronucleus. The diploid micronucleus now goes through mitosis. Once, twice, 3 times. So, once goes through, becomes 2 nuclei, twice, now becomes 4, and then 3rd time, now becomes 8. So we end up with 8 micronuclei inside of each of the 2 paramecia. We're just following one now to keep track of things, but the same thing's happening in the other one. So we've got a macronucleus and 8 micronuclei inside of the cell. The original macronucleus now starts to disintegrate. And 4 of the micronuclei start going through lots and lots and lots and lots and lots and lots of replication of their DNA. That is how they become macronuclei. So many copies of those chromosomes are produced. We end up with the cell having 4 micronuclei and 4 macronuclei. The cell now goes through 2 rounds of cytokinesis, dividing up so that each resulting cell gets 1 micronucleus and 1 macronucleus. Why do they do this? Why are they so difficult? I don't know. But this is how paramecium goes through sexual reproduction. Complicated, convoluted. Why do they need a macronucleus with many hundreds of copies of the genetic material to direct cell activities? I don't know. What's the point of the micronucleus? I don't know. But this is how they do it. Normal everyday function directed by the macronucleus. Micronucleus only comes into play when they're going through sexual reproduction. Weird stuff, unusual, but that's just how protists are.